Unit is another name for cell. So what we're doing is studying single brain cells. So what we want to understand is, basically we listen to the cell. It's firing nerve impulses, and we can turn those into sound and we can listen to them. And we're basically trying to understand the language that they're using. Although we call it single unit physiology, actually we study dozens of these at a time, and as technology is developing, that's becoming hundreds and maybe thousands at a time. So we're starting to build up a picture of the kind of the orchestra, if you like, and what all the instruments are doing. The thing that I've always been interested in is the fine details of how the brain makes knowledge. So I've always thought it's a really interesting question. How do you make knowledge using neurons? And to understand how that's done, you need to study neurons. And so I'm interested in what's called neural coding. How do the neurons represent aspects of the environment or aspects of what's going on? And single neuron recording lets you study in real time the activity of the neurons. So it's not like anatomy or some of the other methods that are slower, or where you're looking at lots of neurons at once. This is very precise. It has quite a few limitations in as much as the technology that's required to look at the activity of single neurons means at the moment that you have to get right inside the brain. You have to put a wire right next to a neuron. And so that limits us to animal studies at the moment, or very occasionally human studies for human patients who have implanted electrodes for clinical diagnostic reasons and so on. But mostly we're restricted to animal experiments, which means that we're restricted to the type of intelligence that animals have. And in the beginnings of this technology, we've only been able to study a few neurons at a time. As the technology is developing, we're starting to be able to record more and more, but still, at most, we can't get more than 1,000 or a few thousand, whereas of course the brain has millions and millions of neurons. So we can get very precise information, but only of a small part of the activity at once. So really, it's only providing one part of the jigsaw puzzle, and we need lots of other methods to try and get at the same issue from different angles. It's not an inherently very amusing subject. There's, I suppose, one thing that stands out in my mind is when I was learning this technique, I had just started in the laboratory of John O'Keefe, who discovered these neurons called place cells. He won the Nobel Prize for that quite recently. And I was in his lab and I was learning how to record, and I spent a happy afternoon recording what I thought was a beautiful cell. And I sat down to look at the pattern of its activity, and I realized that it was firing at almost exactly 50 hertz. And I thought, a cell doesn't fire at 50 hertz, but Maine's electrical frequency is exactly 50 hertz. And so I'd spent several hours recording what we call Maine's noise, and I just had to throw the whole thing away. So I learned a very powerful lesson at that point, to always check the pattern of the activity.